So here we have our DNA and our double helix. So the first step of the transcription process happens when the RNA polymerase moves down the DNA sequence to copy and read the nitrogenous bases to help the RNA version of these bases that are floating around in the nucleus find their match and create the messenger RNA, which is here. And the messenger RNA is the copy of the DNA that will be able to leave the nucleus. And so you may be wondering, why can the messenger RNA leave the nucleus but the DNA can't? Well, there's a few differences between the two. The first difference is that the DNA and the RNA have different sugars. DNA is deoxyribose and RNA is just ribose. The second difference is that RNA has the nitrogenous base uracil that matches with adenine instead of thymine matching with adenine. And the third difference is that you can see it pretty clearly. DNA is double-stranded. It has two backbones when RNA is only single-stranded. And that makes it smaller, which makes it easier for the RNA to leave the nucleus. So here we have our almost finished mRNA. It's done being copied, and next thing that happens is we have some caps that are put on it. We have a 5 cap, and we have a poly A tail. So there's one last step that happens in transcription before the mRNA can leave the nucleus. So in the mRNA, because the nucleus really just does not want to let go of the information, there's a lot of misleading information that's in there. And so all of that misguiding and misleading information is split apart. And there are two things that are left. There are the introns, which are the misleading information, and that gets left in the nucleus to be recycled and reused because it's not useful in our mRNA. And then there's also the exons, which is the useful information that is left in the mRNA outside of the nucleus. There's one more thing to note about DNA before we move on to the process of translation. So every DNA has a specific sequence, and this specific sequence of DNA makes up a gene. What the genes do is they have the instructions to make the molecules called proteins. Now that the mRNA has safely left the nucleus, we move on to the process of translation, translating our genetic information into amino acids and proteins. So here we have our ribosome. Our ribosome is located on the rough ER, and the ribosome is made up of proteins and rRNA. The rRNA acts as a binding site where the mRNA, the messenger RNA, will interact with the tRNA. So there's one thing that happens before the tRNA. So now that our mRNA has left the nucleus, it finds its way to the ribosome. And what the ribosome does is it reads these nitrogenous bases 3 letters at a time, are called a triplet codon. And so what happens is the ribosome will read these nitrogenous bases, and it will find a tRNA or a transfer RNA that matches with the mRNA that has been read. And so the transfer RNA, what it's going to do is it translates the nucleotides into amino acids, which will eventually become proteins. And so there are 2 ends to the transfer RNA. We have one end that has the anticodons. These anticodons are going to match with the 3 codons, the 3 first letters of the mRNA, and each amino acid up here has its own sequence of anticodons at the end. So these 3 anticodons have one amino acid, and a different 3 anticodons would have a different amino acid, and that's always set. And so it reads them, it matches up, and then it'll create that amino acid. It'll move over, and then another 3 codons will be read, and then another transfer RNA will find it, and then that will bind the first one, and then this amino acid chain is going to build on top of each other, and it's going to become a polypeptide chain, which is the beginning of a protein. The last and final thing that you need to know for protein synthesis is the use of the codon wheel. It is used to tell us what amino acids come from the triple codons that the ribosomes read. So UCC would create a different amino acid, UGU. And when those amino acids come together, they create the proteins that are in all of our bodies and make up the muscles and give us energy for living our lives. And that's protein synthesis.